How about now? Am I good? Is it still working? Okay, great. Yeah, so I'll repeat what I said. Um, RNA polymerase will bind to a site at the start of the gene and then will move along it. And the first step is to unwind that section of DNA. Like I said, they usually say that only about one turn of the DNA double helix is unwound at a time. So the RNA polymerase basically works in sections. The next step is that the RNA polymerase will pair up free RNA nucleotides to DNA nucleotides on the template strand. So it'll basically be similar to how replication works, where DNA nucleotides are being attached. Here, RNA nucleotides will be attached to the template strand, and of course, this is done through complementary base pairing, with the bases of the nucleotides linked by hydrogen bonds. RNA polymerase obviously goes along the strand of the template strand of the DNA, and the way it forms a single RNA strand is by linking the nucleotides together as it goes along the template strand. Those links are formed through covalent bonds, or hydrogen bonds. Not hydrogen bonds, sorry. Covalent bonds between the pentose sugar of one of the nucleotides and the phosphate of another. So most of you should know this, that RNA nucleotides and DNA nucleotides have a pentose sugar, a phosphate group, and a nitrogenous base. The nitrogenous bases are ATGC for DNAs, AUGC for RNA, and each of the nucleotide, and then the pentose sugar obviously is ribose for RNA, deoxyribose for DNA, and then there's a phosphate group. So in order to form those covalent bonds, in order to form a single strand of RNA, there's covalent bonds between the phosphate group of one RNA nucleotide and the pentose sugar of another RNA nucleotide. And yes, it does contain uracil. I will get to that in the next slide. So the assembled RNA strand, after the RNA polymerase has moved across the template strand and is done with transcribing that specific gene, the assembled RNA strand will separate from the template DNA strand, and the DNA strands will then, since they're free, will rewind back into a double helix and then just become normal again, as if nothing happened. Does everyone understand how transcription works? Is there anything that doesn't make sense? Is there anything I said that is confusing? Are there any questions? Is replication the same as translation? No, you don't have to save questions for later. I'm like, if you have general questions about not transcription, then save them. But if there's questions about transcription in specific, I will be able to answer them right now. Anyway, why does our body do this? So the reason transcription is important, and I will talk about this again later, is for protein synthesis. After transcription is what allows our body to synthesize proteins to express the genes, essentially. The purpose of transcription is to express genes in our DNA into messages that other parts of our body can read, essentially, which I will explain again. So if you know what transcribing is, ligase doesn't play a role in transcription, no. It's basically metabolic functions. Basically, yes. The point of transcription is to make it easier to read, essentially. So if you know what transcribing in general means, people will transcribe their handwritten work onto a digital document. That's transcribing as well. So things like that, things that make it easier to read. So essentially, transcribing in our body as well is to make the gene sequence easier to read for other parts of our body through the mRNA, the RNA strands that are created. If that makes sense. Is that a good enough answer? Not sure, but I hope that makes sense. mRNA I'm going to talk about more later when I'm talking about translation. This is just, we're just talking about it being just it's just forming an RNA strand. Protein synthesis allows for gene expression via mRNA to genes in DNA. No, that's not how it works. Basically, the genes are what code for certain proteins, right? So transcription is to allow those proteins to be coded from the genes that are present in our DNA. Our DNA has certain genes. Those are transcribed through transcription through RNA polymerase to form an RNA strand. That RNA strand is then interpreted by our ribosomes, which then create protein polypeptides. If that makes sense. I will talk about more of this later. I will come back to it. Okay. Hydrogen bonding. So we should all know this by now, but I'm going to repeat it anyway. So the bases in DNA molecules in general are linked. Obviously, ATGC, adenine and thymine are linked together, and cytosine and guanine are linked together. However, when we're going from DNA to RNA, there is a change, because in RNA, there is no thiamine. So in order to accommodate for that, the bases in the RNA and DNA will still pair up with hydrogen bonding, but the way they do it is different. So adenine and uracil, cytosine and guanine are the 4 RNA bases, but when it's transcribing from DNA to RNA, it's going to go adenine to uracil, cytosine to guanine, guanine to cytosine, those are normal, and then the thiamine will become adenine. So then the thiamine just doesn't exist in the RNA strands. The reason hydrogen bonding is used in this case is because hydrogen bonds are fairly easily broken, which is important because the RNA transcript, or the RNA strand that has been transcribed already, needs to be separated from the DNA template strand, and in order for that to happen, the hydrogen bonding has to be broken. And then once that hydrogen bond is broken and the RNA strand leaves, then there's hydrogen bonding that happens again between the DNA strands. So another piece of terminology, there's the template strand, and then the strand opposite to the template strand is the sense strand. So when transcription happens, the mRNA is going to be pretty much identical to the sense strand, with the only difference being the thiamine is uracil in the RNA strand rather than the thiamine in the sense strand. Does that make sense? The what? Sense, SENSE, sense strand. So DNA has 2 strands, right? The template strand is where the mRNA is constructed, and then the strand that is not being used is called the sense strand. So SENSE, sense strand. That sense strand will be identical, like the base sequences in the sense strand will be identical to the mRNA strand, with the exception being that in the sense strand there's thiamine, and in the mRNA strand each of those thiamines actually is a uracil instead of a thiamine, because RNA doesn't have thiamine, RNA has uracil. sense and antisense instead? Technically, no, because you have to say that it's a template strand. It is sense and antisense, but you have to use the fact that it's a template strand because that's where it's being made. I would recommend you just say template strand. But it is technically, yes, it's sense and antisense. Template and anti-template, no, because it does not. Unfortunately, that's not how it works. As long as, actually, I don't think they'll mark you down if you say anti-sense either, but you just have to know that anti-sense is where the template happens. But otherwise, if you use anti-sense, I don't think they'll mark you down for it. Anti-template is not a actual term. It's not accurate terminology, so I wouldn't say that you can use it. Template strand is the strand used for coding mRNA, and sense strand is not used, but sense strand and mRNA, the mRNA strand and the sense strand will basically be identical with the exception of thymine and uracil. Cool. Okay. So, conservation of DNA templates. Now, the components of DNA, so there's phosphate, deoxyribose sugar, and the four bases ACT. I don't know why they do ACGT, but it's ATGC in my head, are all really stable components, right? And they're linked together in a strand of DNA by strong covalent bonds. The sugar and phosphate groups form the backbone to which the bases are attached, and this stability is important because this stability is important because it ensures that the base sequence of a gene will rarely change during transcription. The stability is formed through the covalent bonds. The covalent bonds are really, really hard to break, which makes it really, really stable, which means that the base sequence will not be changed. If the base sequence changes, that changes how the gene is expressed. So a gene can be transcribed as many times as necessary, but it doesn't change between transcriptions. Does that make sense? This is basically just a point to remember. Like, it can be something like, how is the integrity of the gene, or the integrity of DNA template preserved? And the answer would be something like the covalent bonds create a strong backbone to which the bases are attached and cannot be removed easily, which allows for consistency between transcriptions. Okay, does that make sense? Yeah, mRNA is created on the antisense strand, not on the sense strand. Okay, gene expression. So this is where your answer should be answered more or less. So, a few things. A gene is expressed when the information it holds is used and an observable characteristic is generated within a cell or an organism. At any specific time, there are some genes that are being expressed and most are not, because there's very specific sections of genes that need to be expressed in very specific cells, because cells are specialized in our bodies for particular functions, and they will only develop the characteristics that they need. They will not express genes that they do not have use for, essentially. Transcription is the first stage of gene expression, and it can be switched on or off, which is really, really important because that's how gene expression is controlled in our body. So transcription only happens when that specific gene has to be expressed in a cell. Transcription will not happen if the gene does not need to be expressed in that particular cell. So the way gene expression works is that there's the DNA molecule. The DNA molecule will be transcribed into an RNA copy of the sense strand's base sequence. That RNA copy will be moved to a ribosome where translation will happen, and that RNA sequence will be translated into an amino acid sequence of a polypeptide. That polypeptide will then be used their structured polypeptides which will be the direct cause of observable characteristics, but then there are also polypeptides that will be used as enzymes in the body. So the entire point of gene expression is to have this, like, to create this polypeptide, which will then either be used for certain characteristics or will be used as an enzyme in the body. So it's either metabolic processes or just for the observable characteristic part of it. Do people understand why transcription and translation happens in the body? Does everyone understand gene expression? And does this kind of answer your question, B? Because if it doesn't, I can try to re-explain it. Okay, I will finish this and then you can ask me more questions. Okay, main points and differences between translation and transcription. I have yet to explain what translation is, so let me do that first and then we can talk about the differences between translation and transcription. I haven't explained what translation is yet. This is just the most general, like, terms, just for you to understand how gene expression works in our body. Yes, that's a pretty good description, I would say. Yes. The process by which information that is encoded in the DNA, quote unquote, the gene, is used to direct the synthesis of a protein molecule, which then can be used for either metabolic processes or observable characteristics. Okay, now we're going to talk about translation. Translation is the synthesis of a polypeptide with its amino acid sequence determined by the base sequence of an RNA molecule. Now, that RNA molecule comes from transcription. That's how they're linked. The RNA molecule is transcribed from a protein encoding gene and is called messenger RNA. Not protein encoding, protein coding gene, and it's called messenger RNA. So mRNA is mRNA is the thing that is then translated into polypeptides of proteins. Each base in the polypeptide is coded for by one codon on the mRNA, and a codon is a sequence of 3 bases. So essentially, each mRNA strand will have multiples of 3 bases, and each 3 sections will be coding for one specific amino acid. So as you can see at the bottom here, for example, GCU is one of the 64 different codons, but one of the codons, and that codon will then translate to a specific amino acid. And we know of 20 amino acids in IB biology, and so this 3 will then code for one very specific amino acid. I mean, these 3 bases. So it's guanine, cytosine, and uracil together in that sequence will translate into alanine, for example. And then there's tRNA. So I just explained what mRNA is. This is tRNA. So tRNA is transfer RNA. This is a group of RNA molecules which essentially allow for amino acids to be transferred. Let me use the words that isn't there. tRNA is transfer RNA, and they transfer the amino acids in order to be translated. So there's still codons. So 3 bases at one end of a tRNA molecule bind to a codon on the mRNA during translation, following the rules of complementary base pairing. So this is where you need to know. So tRNA has anticodons and mRNA has codons. Each of the tRNA's anticodons has a complement to the mRNA codons. And then at the other end of the tRNA molecule is a point where amino acids can be attached. And that's where the polypeptides can be created, because each tRNA molecule has a link to a specific amino acid, and then that amino acid is attached. When I explain translation better, give me a second, sorry. A lot of this will not make sense until I actually explain the translation process, so just bear with me for a second. There's a few questions I will check. RNA from the transcription is called mRNA in translation. So transcription will create RNA strands. Specific RNA strands that are used for protein synthesis are called messenger RNAs. And those messenger RNAs are used for translation. Yeah. So, like Fauna said, the RNA from transcription is mRNA, which moves into translation. Yeah. So each tRNA anticodon each tRNA has a group of 3 nitrogenous bases, and each of those 3 groups is an anticodon because they're the complements of the codons on the mRNA. So for example, let me go here. So this is a codon on the mRNA, right? So the link to this codon on the mRNA would be an anticodon on the tRNA, which is CGA. So this is the mRNA codon. This is the mRNA strand. This codon has an anticodon on the tRNA, which is what determines which amino acid is linked to it. Which is why when we say that each codon codes for one amino acid, that's because each codon has a specific tRNA that links to it, and each tRNA has a distinctive shape which links to specific amino acids. Let me explain this when I'm talking about translation. But before we do that, we have to talk about ribosomes. Sorry if it's a little bit scattered. I had to explain what things are before I start explaining translation as a whole. But it's a little bit shuffled, if that makes sense. Anyway, so ribosomes are where translation happens. Everyone should know what a ribosome is. Ribosomes are where translation happens, and ribosomes have 2 subunits. There's the large subunit and the small subunit. The small subunit is where the mRNA binds, and the large subunit is where there are binding sites for tRNA so that they can attach so that the amino acids can be attached to each other there. The rest of this I will explain when I'm explaining translation. Okay, so for it to make a little bit more sense. This is the mRNA strand, okay? This is the mRNA strand, and it's moving in this direction. So this is, for example, a codon. These 3. These 3 are a codon, and then these 3 that you can see over here are a codon, and these 3 here are a codon. Now, each of these codons have an anticodon on the tRNA, and it's very specific anticodons. Each tRNA can only bind to one amino acid, which basically means that each codon determines which tRNA comes and binds to then supply this specific amino acid. So the way translation works is that the mRNA will bind to a site on the small subunit of the ribosome. This is the small subunit of the ribosome. And the mRNA has a series of codons. So like I said, codons are 3 bases. So that's a codon. Those 3 are codon. The 3 that are linked to this tRNA are codon. These 3 that are linked to a tRNA are codon, and so on and so forth. Each codon codes for one specific amino acid. tRNA molecules are present around the ribosome in large numbers. Each tRNA has a specific triplet of bases, which is called the anticodon, because it is complementary to the codons on the mRNA strands. Now, each tRNA will carry a amino acid which corresponds to the anticodon. So it's very specific. Each tRNA will only have one amino acid to bind to. Those tRNA molecules have binding sites in the large subunit of the ribosome, and those binding sites are where the tRNA will arrive at, essentially, and then link to the mRNA at the small subunit. A tRNA can only bind if it has the anticodon, which is complementary to the codon of the mRNA. The tRNAs that don't have the specific anticodon will not be used. They'll only bind if they have the specific complement in the mRNA. And then, again, like I said before, there's hydrogen bonding. They are still the same bases. So then the anticodons and the codons will be bound by hydrogen bonding with the same rules of complementary base pairing. And then the amino acids that are being carried will be linked together by peptide linkage, where everyone should know what a peptide bond is by now. I don't want to have to explain what a peptide bond is. But basically, peptide bonds are formed between each amino acid and then forms a long chain of polypeptide. That polypeptide is then used as a protein. At one point in the large subunit, there's only 2 tRNAs present. And what happens is that, for example, this tRNA has arrived and this tRNA has arrived. The dipeptide forms between these 2 amino acids and then basically get attached to only this tRNA molecule, and then this tRNA will leave. And then the ribosome will move along the mRNA to the next codon. So basically, after this one, it'll move to the next one, where another tRNA will come and bind, and then that amino acid then attaches back to the polypeptide, and so on and so forth, until a full polypeptide is formed and the entire mRNA is translated, is the word. Sorry. You should know what peptide bonds and condensation reactions are. I don't think that's part of this. That's part of... What part? What? It's part of nucleic acids or something? It's between... No, it's in the protein chapter. It's in sorry, I don't know how the new, I still don't know what the new syllabus, like, subheadings are, I'm sorry. But it should be with the protein and enzyme subtopic thing, whatever it's called. Yeah, I don't remember the numbers and everything, so yeah. Okay, direction of translation and transcription I'm going to talk about later because that's an HL only thing. So I'm going to hold off for a second. Sorry. Okay, does everyone kind of understand translation? Sorry, I know that it was a very scattered explanation of what translation is. But if there are questions, ask them now, because the better I can try to re-explain it, the better it will be for you as well. So ask questions. Please ask questions. This is where I'm like, please do ask questions. What was the thing in the ribosome? So each ribosome has 2 units. There's a larger subunit and there's a smaller subunit. The small subunit is where the mRNA will be attached, and that's where the ribosome will move across. The large subunit has 3 binding sites, but out of those 3 binding sites, only 2 are occupied at the same time. But those binding sites are for tRNA molecules with their amino acids. I know what you mean, and I have a question for that after this slide, I think, or the next slide. I can't remember. No. There. We're going to talk about coding in a second, in a bit. That's next. I'm going to talk about degeneracy and universality next. And coding after I talk about degeneracy and universality. That will be after that. I'm just talking about translation as a process. Apparently this is also SL. I don't know. It says it on, again, I'm following the IB study guide, and on the study guide it's put as SL still. So I just keep going. If it's only for HLs, I apologize to the SLs. Okay. Movement of the tRNA. After the anticodon bind to the codon, we have amino acid after condensation, right? Because the large subunit, there's only space for 2 tRNA. Okay, let me try to explain this again. Basically, this is where it started, right? So the ribosome is moving this direction. Each large subunit of a ribosome has 3 binding sites. Out of those 3 binding sites, at one point only 2 tRNA will bind, but essentially what happens is that when those 2 bind, a peptide bond is created. There's a dipeptide linked to this one. This will detach the let me find you a better image, actually, showing the EPA sites and everything. Is the EPA sites only HL? Sorry. EPA sites is only HL. But I think in general it might give you a better, and I think the people asking the questions are HL as well, so it's okay if I do a little bit of a, here, actually, this might be a good enough. And this doesn't have EPA sites or anything. Okay, give me a second, guys. Let me just, this is not a bad diagram. There's also let's see. No, hold on. Yeah, sorry. I can't. Thing is, but like for HLs, for SLs, if you don't need to know it, I feel a little bit bad trying to do it like that. But anyway, okay. I'm gonna use this. Let me go back. Okay, so basically, what happens is this ribosome is moving in this direction, okay? So, for example, this tRNA molecule is here, this tRNA molecule is here. It attaches, and then, this amino acid gets attached to the polypeptide chain, and no, that's a lie. Sorry. This polypeptide chain and then this amino acid get attached together. The polypeptide will be attached to this tRNA molecule. This tRNA will leave. The ribosome will move across. This new tRNA will come join over here. And then the same thing repeats itself. So the polypeptide chain or the amino acid chain is moving towards the right as the ribosome is also moving towards the right. Does that make sense? Okay, are there any more questions that I can help with? What do you need to know about lac-operon though? Oh, I might explain it later then. Actually, never mind. Interesting. But what do you need to know about lac-operon? I don't... Okay. Sure. Polysome a release factor, like... Anyway. Yeah. It's... Okay, so the ribosome is moving from left to right. And the... the peptides will always attach towards the right. So if it's going from left to right, so are the peptides moving from left to right. It will move in the same direction. Does that make sense? Because the point is that in order for the ribosome to move, you would still have to have the anticodon that connects to the next, like to the middle codon, right? If it goes from here to here and this one detaches, then you can't move across because this anticodon would not mix with the next codon, if that makes sense. Like the point of it going from it all attaching to the right side is that when you move the ribosome here, you're still gonna have the polypeptide attached to a tRNA that can actually attach to the next codon. If you do it to this one, then it's not gonna attach to the right, like, then if you can't move across because this tRNA can only attach to this codon, not the next codon. Does that make sense? Yeah, E's for... No, so, okay, you have that right. E is for exit, but the exit is for the tRNA molecules, not for the amino acids. The amino acids will attach to the right. they attach to the next, because you need to keep the amino acids together because they're a single chain, and you need to attach the next amino acids back to the thing. The amino acids will only quote unquote leave once the entire mRNA translation is done. So when you reach the end of the mRNA strand, then it'll leave together with the last tRNA molecule. Like, it always has to be attached to the middle. Like, the polypeptide always has to be attached to the middle thing. That's when the ribosome, like, detaches from the mRNA. Yeah. So that's fine. You can do that. It always has, the polypeptide always has to be in the middle, because in order for it to move across the mRNA molecule, and the new one always comes through. It's more so the tRNA position, but like if you want to know, I mean, like, the amino acids will be in the middle, because in order for it to move across the, what's it called, freaking mRNA molecule, you have to have it in the middle so that the new tRNA can come and the old tRNA can exit. Okay. Anyway, this EPA stuff is more HL anyway, so I will explain that when I'm talking about the HL parts. But in general, do people understand how translation works? Did my previous explanation make more sense than my first one? Because my first explanation was not great, but we tried. We move. Oh, people are writing questions. Okay, I'm gonna wait. This is from the study guide. All of my notes are from the study guide, bro. tRNA attaches with an amino acid. Okay, again, we're using APE sites, and I don't want to talk about APE sites right now because that's HL specific. Okay, I will talk about that when I talk about the EPA sites. Hold on to that question, and I will answer that when I talk about EPA sites. Because I don't want to talk about HL stuff in the SL section, because then the poor SL kids are going to be like, what are you talking about? And so I don't want to do that. I don't know, whoever is the SL kid. Okay. What was the question? Yeah, that's, I think that's still HL, if I remember correctly. Because that's, the initiation and stuff that's all HL. That's not SL. That's still HL. Yeah. Elongation is where I'm going to stop for SL. No, there's mutations as well. Mutations is where we're going to stop for stuff like that. Okay. Anyway. I'm going to continue. I think we're done. Yes? Okay. Good. Next. So, genetic coding. We're going to talk about the fact that it's in triplets. We're going to talk about degeneracy and universality. So, genetic code operates in triplets. So, as we talked about, there are codons, which consist of 3 bases, which code for one specific amino acid. So, it's a triplet code, which makes sense. Three bases per thing. With 2 bases, the reason it's in triplets is because with 2 bases, there would only be 16 codons, which would not be, that's not 4, hold on. It's because my freaking, let me, hold on guys, sorry, no, I freaking hate this laptop, kill me, no, kill me, okay, 4 squared, no, okay, give me a second, I realized that's, there we go, okay, yeah, so with two bases, there would only be 16 codons, because 4 squared, which would not be enough for the 20 amino acids. With 3 bases, there are 64 different codons, 4 to the power 3, which are more than enough for the 20 amino acids in polypeptides. The reason it's 4 to the power 2 and 4 to the power 3 is because there are 4 bases, and 2 and 3 are how many are in each codon. I hope you guys understand that, and I don't have to re-explain that. Okay, degeneracy. So there are more codons than the minimum needed to code for the 20 amino acids. As we said, there are 64 different codons and we only technically need 20. However, even though there are more, none of the 64 codons are unused, and instead there will be 2 or more codons for most amino acids. Most amino acids will have at least 2 codons that code for it, or sometimes even more. So a code in which more than one symbol is used to represent the same thing is known as a degenerate code, hence why its degeneracy is a characteristic. Next, universality. So, give me a second. The 64 codons of the genetic code have the same meanings in cells of all organisms. Apart from a few minor variations, they basically have the same meanings in every organism. Which basically means, well, not means, but universality of the genetic code is strong evidence for all life on Earth having evolved from the same original cells, with the minor coding differences accruing since this common origin, or basically just changing since the origin. Does that make sense, guys? Do we understand triplet degeneracy and universality? Okay, I will wait. If there's a question, then yes, I will wait. T, you already know this, so explaining this to you doesn't make sense. Okay, so there is the degeneracy part. So a degenerate code is basically when more than one symbol is used to represent the same thing. More than one symbol is used to represent the same thing. So as we know, there are 64 codons rather than just the 20 that we technically need for it to be 1-to-1 ratio with the amino acids. There's 64, which means that more than one codon represents the total. Like there's multiple codons that will represent a single amino acid, and that's why it's called a degenerate code, and that's why degeneracy is a characteristic of our genetic code, because there's more like, does that make sense? I don't know if that made sense. I just repeated everything I said before, but if there's a more specific question, you can ask me. Amino acids significant. It's not. It's just a characteristic of the genetic code. It's not really a like significant thing. It's just one of 3 characteristics that you need to know. So they'll be like, just like what are characteristics of our genetic code, and then yeah, it's just this exists, no this exists type thing. Does that make sense? Good? Are we good? Yes? Yes. Okay, perfect. We're talking about elongation now. I didn't add a thing on this because I just literally just stole this from the study guide because I cannot explain this in any other way. So yeah, there's no title or anything. This is just gonna be yap. So I'm gonna start at the top. We're gonna start at the top. It's basically a cycle, so you can start anywhere, but we're gonna start at the top left. So as we know, a peptide bond is made between the amino acid. Okay, by the way, this is in the SL portion, but it says EPA, which I'm like so confused about, but I'm just gonna talk about it anyway. This is like, it's pissing me off how they're doing this because it literally says SL, but it also says EPA thing. But anyway, I'm not stealing, I'm just borrowing, guys. It's fine. Anyway. Yeah, so a peptide bond is made between the amino acid held by the A site tRNA and the amino acid at the end of the polypeptide held by the P site tRNA. So for SLs who don't know what EPA sites are, I'm just going to explain it just for context. There's the E site, the P site, and the A site, and they're in that order. Something to remember, I guess, would be the E site is quote unquote the exit site, and then P and A are just, like you can say A is the attachment site, whatever. You can find ways to remember this better, but basically there's E sites, P sites, and A sites. And those are the three binding sites in the large subunit of the ribosome. So the direction of the ribosome is in this direction, right? The way it works is this tRNA, for example, already exists. Actually, I should start here probably. That's probably a better... No, hold on. We'll start here. Never mind. Because they've arranged this in such a confusing... Anyway, let's not. I'm getting mad at a textbook right now. Anyway, so we're going to start here. Basically what happens is a tRNA molecule with an amino acid will come into the A site, attached to the codon on the mRNA using hydrogen bonding. So, for example, here, a tRNA with the anticodon complementary to the next codon on the mRNA will bind to the A site. The tRNA at the P site has the polypeptide that has been so far assembled. So in the beginning, it'll just be one, but after that, it'll be a polypeptide that has been assembled so far. So the new tRNA will come and join in the A site, and then a peptide bond will be formed between the amino acid on the amino acid on the tRNA on the A site and the polypeptide on the tRNA on the P site. There's a peptide bond formed between them. When that peptide bond is formed, the polypeptide from the P site will be transferred into the polypeptide on the polypeptide on the tRNA on the P site will be transferred to the tRNA on the A site. So this polypeptide will move here. So as you can see in this one. And then the ribosome will move 3 bases along. So these 3 bases are then sacrificed basically. And then we start here and move along. And then the next 3 bases are vacant. And then when it moves that way, basically this tRNA will move to the E site, the tRNA with the polypeptide will move to the P site, and then the A site will become vacant again. The next step is the tRNA that has now freshly moved from the P site to the E site will then leave the ribosome, which is why E is for exit, will leave the ribosome, and the E site will then be vacant again, and then it continues the cycle again where at the A site the new tRNA comes and has the anticodon that is complementary to the next codon, and then so on and so forth. So that's how the changes happen. This is technically still SL, even though I feel like it should be HL, but this is technically still SL. It's called elongation of the polypeptide chain. Okay, does this help a little bit more? This is technically, in my opinion, very much also HL. Like, this should hopefully make more sense be about your EPA site confusion. So essentially what happens is we start here where the polypeptide is currently on the P site, then a tRNA will come to the A site, there will be linkage happening, and then the P site tRNA will then be empty, the A site will have the polypeptide, the ribosome will move, and then we'll go back into the polypeptide being in the P site, and the E site will have the E site will have the empty tRNA, which then leaves from there. Yeah, I did say that, but yeah, A for arrival and E for exit. So A is where the new tRNA comes in, E is where the old tRNA will exit from. Okay. Whew, does that make sense? That was a lot of yap. Anyway, does that make sense? Are there any questions? Actually, should be my thing. Just think AP. I think EP usually. EPA. In my head, I just think EPA, but I don't know. Yes, I can. Okay, so step 1. A tRNA with the complementary anticodon to the codon of the mRNA binds to the A site. A peptide peptide bond is formed between the existing polypeptide on the P site tRNA to the amino acid on the tRNA on the A site. That peptide bond creates a polypeptide which attaches to the tRNA in the A site. That's the next step. Step 4 is the ribosome moves across 3 bases, or basically a codon, and moves a codon's length to the right. When that happens, the tRNAs then move their binding sites from P to E and A to P, which means that the empty tRNA is now in the E site and the polypeptide tRNA is on the P site. And then the empty tRNA will leave from the E site and the polypeptide will exist on the P site until the process happens again, where a new tRNA arrives, etc., 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 etc. Yes. It's always EPA, which is why in my head I remember EPA. But sometimes some people will prefer to remember APE because that's the direction in which the tRNAs come and leave, because it goes APE, like they go from, the tRNAs will come in through the right and leave through the left, although the direction of the ribosome moving is from left to right, and the direction of the sites is EPA. But you can choose to remember it however you think you'll best remember it. Okay. Yes. Okay, no, even direction-wise, depending on what you're saying, direction of tRNA movement is from APE, but if you're talking about direction of movement on the mRNA, it's from left to right. Just make sure you don't mix, just make sure you don't mix them up, because then otherwise they're going to be like... you're wrong type thing. Okay. Hopefully that makes more sense in terms of translation, because I know the way I explained translation first didn't make a lot of sense, but this will hopefully clear it up a little bit more. I hope. Okay. Questions? No questions? Why am I going backwards? I will drink some water while people type questions if they have any. To be fair, I'm running quite good on time in comparison to other sessions that I've done. Mentally beginning to die. You're so real though. That's so real. Okay. I'm gonna wait to see if there's actually a question, and then we will. Six mark about translation and transcription. Am I allowed to draw a whole diagram? Yes, you can. Absolutely. As long as you're still explaining it, then absolutely yes. Go ahead, draw a diagram. Go for it. Transcription translation is protein synthesis. Yes. Congratulations. Replication is to create new DNA molecules. That's not related to transcription translation. We didn't even used to call it protein synthesis. We just used to call it transcription and translation in the old syllabus, if I remember correctly. It was literally just called DNA transcription and DNA translation, if I remember correctly. But now it's called fancy protein synthesis. Back in my day. That's what I'm saying. Back in my day, bro. A year ago. Okay. So, we're going to talk about coding. And then we're going to talk about mutation. And then we're going to be done with SL, I think. So, we're almost there, SLs. Hang on. HLs, I'm sorry. I'm just sorry. I don't even know what to say. I'm just sorry. Transcription is only copy paste onto mRNA translations, finding mRNA using link to amino acids together to. Yes, absolutely. Mutations is not. No. The mutations I'm going to talk about right now is not the D1.3 mutations. This mutations is just like the basic, like protein synthesis mutations. It's very basic. But yeah, Laura, you're right. That is, yeah, that's accurate. Okay, let's talk about coding. So, as we talked about, there's codons that each code for a specific amino acid, right? Now, there are certain amino acids. There's certain codons that code for stop signals that indicate when the synthesis of the polypeptide is meant to end. There's one codon that represents both a start signal and also the specific amino acid. Methionine, I can't say it. Whatever. That thing. Or MET. That amino acid is the start signal and is also the first amino acid in every chain of polypeptides. You don't need to remember the table. The table is usually given to you. But you should know methionine. And you should know that there are stop codons and there are other amino acids. But you don't need to remember it. But yeah. Okay, so a little bit of an exercise for you guys. Translate the following sequence. It's only 4 different. Yeah, that might be a little bit better. I don't know. I just genuinely just like copied it from the the fuck is it called? Study guide. But that, I like that picture actually. I might add that, honestly. I can't lie. Anyway. Yeah, translate, guys. Go on. Do it. Please spoiler your answers, actually, before you start answering the questions with the enthusiasm of M25s. Just remove where I say text. How do you use this table? So, on the left is the first base of the codon. On the top is the top base of codon, and on the right is the third base of codon. You can also use the table that Lori sent. That might be a better one, but like in the exam they won't give you that one, so unfortunately it won't work like that. But basically what happens is, for example, this first one is U, and U, and that translates into phenylalanine. Phenylalanine. The fuck am I saying? Phenylalanine. Whatever that is. And then U, C, and U would be serine. U, for example, C, A, G here is glutamine, because it's in the C row, the A column, and then in the G row of the C row, basically. There's 4 bases on the right side because, for example, you can go U, 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 you can go U, U, C, U, U, A, U, U, G. So it can be any of the 4. Okay, I mean, the answer is on the thing, but everyone looks right so far. I will put the answer up in a second after. I don't think anyone else is doing it. Okay, so that's the answer. So I think most of you did it right. It's histidine, and then, yeah, exactly. The one you sent is really pretty and it does everything for you, but they won't do that for the exam, unfortunately, because they are haters. So yeah, anyway, that's the answer. So the way it works, for example, is CAC, right? So C is, it has to be in this row, A, it has to be in this column, so this square, and then C is the next one, so it's histidine. So CAC will be histidine over here. Next is AGA, so AG and A will be over here. It will be arginine, I don't know. And then UGG, so U over here, G over here, G over here, so tryptophan, and then valine, GUC, which you go G, U, and G, U, and any, well, if it's G and U, any of them will be valine, so you can just do that automatically. You don't even have to worry about the C. Does that make sense? Paranoia time. Okay. Go on. So what you were doing was okay, sometimes they can ask you, for example, let me go back. Sometimes they can ask you what was the template strand. So what you just did there is template strand. So you did transcription. I was making you do translation. Do you need to remember the short forms? No, you don't. Why would you need to remember the short forms of anything? They give you the names of the amino acids. You don't need to remember the short forms. They'll literally give you that table, so they have the names of the like amino acids. So you're fine. No, they're not like, okay, the IB is bad, but they're not like that evil where they'll give you only the short forms. But yeah, so B, what you were doing there is like reverse transcription. Yeah, reverse transcription. So you, for example, in like mocks, they might ask, what is what was the template strand, and then give you that sequence. Sometimes they'll even ask you what is the sense strand, and then basically it's the same thing, but you replace all the U's with T's. They would also, yeah, I did the exam last year. They can also, I mean, yeah, so basically what you did was transcription and I asked for the translation. That's it. That's the only difference. But otherwise, technically you still did it right. I'm not a genius. I did bio list. When you're transcribing though, yes, the template strand has to be DNA. So you do T. Binds to U will be still A. It'll just be A. So I said it in this one. Let me go back. Where is it? Yeah. So you have to do the reverse, right? So from DNA to RNA, it'll go from adenine to uracil. So from RNA to DNA, it'll go from uracil to adenine. U replaces T's. The U's replace the T's. In DNA, there's adenine, thymine, cytosine, guanine. In RNA, there's adenine, uracil, cytosine, guanine. That's it. That's the only difference between RNA and DNA. Nucleotides. Okay. Do we understand coding, guys? Are there questions? Oh, IDK. Mock number. Just send the question. Just like take a screenshot of the question. That's it. So this is reverse transcription. Yes. So you're going from the mRNA to the DNA template strand. You're not going from DNA template strand to RNA. Okay. So the table shows the mRNA codons for 3 amino acids. Which substitution mutation of a base triplet? Okay, I'm going to talk about substitution mutations literally next. So let me talk about this first, and then I will talk about the answer in a second. Let me talk about what they are first, because I haven't explained that yet. So let me do that. Okay, so protein structures can be changed by mutation as small as a single base change, what is called a anyway, what was I gonna say? I forgot what I was gonna say. It might be, but don't, please. Protein structure can be changed by a mutation as small as a single base change. That single base change is called a point mutation. So when there is gene coding for the protein going on, that single base substitution can change the codon and therefore will change what protein it is, what amino acid is linked, and therefore change the entire protein structure. So a single base substitution is when one codon in the mRNA is changed. Well, a single base substitution, a point mutation and a single base substitution is the same. A single base substitution is when, for example, instead of a G, a T gets added. That base substitution will change the codon in the mRNA, which will then change the amino acid in the translation, which will then change the entire structure of the protein. Single base substitution is a type of point mutation. So, like I said, even a single changed amino acid can change the entire protein structure, especially if the substitution is between hydrophilic and hydrophobic amino acids, because if a protein is meant to be hydrophobic and you add a hydrophilic amino acid in it, it changes the entire structure of that protein. In contrast, some base substitutions will have no effect, either because they are same sense mutations. So as we know, there are multiple codons that will code for the same amino acid. So if that, for example, if the mutation is from CUU to CUA, for example, it's going to result in the same amino acid, which means that it's a same sense mutation. So the codon is changed into another codon for the same amino acid. So there will be no effect from that, obviously, because it's the same amino acid. And then there is another way the base substitution will have no effect is when amino acids are very chemically similar to the first one and is in a non-critical part of the protein structure, which means that the protein structure will not have changed enough for it to be an issue. So that's what mutations are like. Okay. An example is the sickle cell mutation. So HBB is the gene that codes for hemoglobin, which consists of 146 amino acids. Now, in those 146 amino acids, a base substitution in sickle cell mutations, a base substitution mutation will change the adenine to thymine in the codon for the 6th amino acid. This changes HBA to HB, just to HB. Is it just to HB? Hold on. I have it. I copied it down wrong probably. Let me reread it. Yeah, it changes HBA into HBS, which is a sickle cell allele. And basically what happens is that change will result in a different, different protein, and therefore will change how it is coded. No, I'm doing the wrong thing. That's why. HBA and HBS. Okay, there we go. With major consequences. So for example, here, this is what HBA is, which is the normal allele of the gene, right? So the CTC and the G will transcribe to GAG, and then that GAG will then result in glutamic acid, which will result in normal hemoglobin. With a mutation, the HBS will be GTG instead of the normal GAG, and then that GTG will that GTG will have a CAC codon as its template, and then that mRNA will then result in a GUG strand rather than a GAG strand. So that GUG will then translate into a valine instead of a glutamic acid, and that valine will then result in sickle cells, because valine is hydrophobic and glutamic acid is hydrophilic, and then because it has a hydrophobic you're putting in hydrophobic structure in blood, which is a highly like water substance, highly like water substance. I don't know what I'm saying. It has a lot of water is what I'm saying. When you put a hydrophobic substance in a place where there's a lot of water, of course it's gonna go into it, essentially. Aqueous, whatever. There's a lot of water in our blood, we know that, which results in sickled cells. So it is a mutation. Okay, now everything after that is HL. So before we do that, I'm gonna look at this question and try to answer it. Let me just add a slide so I can put it in here so everyone can see. Okay, so we're talking about this question. The table shows mRNA codons for 3 different amino acids. Which substitution mutation of a base triplet on a DNA strand will lead to the same polypeptide being formed at translation? Oh my lord. Okay. So, base triplet on mRNA codons for 3 amino acids. Okay, so the way I would tackle this question is find the opposites of each of these codons. So, for example, GUU will be T, not T, what am I saying? CAA, and then this will be TGA, etc., etc., etc. Now, the substitution mutation that will lead to the same polypeptide is, let me see. I'm trying to do this in my head rather than doing it on my... I might have to use paper and pencil, guys. I haven't used a pencil and paper in like ages. TGA, TGA, TCA, TGA, TCA. Wait, why am I overthinking this? Hold on. G8TCA. That will result in ACT. Yeah, no, don't translate the whole table. Sorry, I said it wrong. Translate the options, not the whole table, because that's just double work, because then you're gonna have to reverse it. You would translate these and then do it that way. Return. Okay, can't be this one. Yeah, so it would be it's basically base pairing. So GCT will be CGA and GGA will be CCG, and then CCG and CGA are both under the same amino acid, which means they will code for the same amino acid. So the way you would work it out, for example, is translate each of these codons. Transcribe each of these codons, because take it that these are codons on a DNA strand, right? You want to transcribe this into mRNA strands, and then that mRNA codon will associate itself with a specific amino acid. Yeah, no, you can't do it in your head. I think write it down is always better. Doing this in your head is more confusing than writing it out. Writing it out is always better. So I would say, for example, like I said already, transcribe all of the options into the mRNA codons and then match each of those mRNA codons with the amino acids. And then you will find that in D, GCT and GGA will result in CGA and CCG. No, CCU, which are both here. So CCU is under proline and so is CGA, which means that they will both result in the same amino acid. Does that make sense? Not translate, transcribe, please. Sorry, translate to the same. No, actually, you're right. You're transcribing the DNA codons that are given in the options and translating them into the codons that match with. Translating them into the amino acids. There we go. That makes sense. T is attached to adenine A's, and A's are attached to U's when you're doing transcribing this way. Also, you're in M26. Have you even studied this yet? Have you studied this yet? That's early days. Oh my lord. Okay. Anyway, does this make more sense? Who asked this question? There was someone who asked this question. I don't even remember who asked this question. Jess, does this make more sense? Okay. Perfect. Okay, that makes sense. Okay, cool. So we're gonna take a little small break here. Because this is where SLs end. Okay, I'm gonna. Okay. Oh wait, let me just. Okay. Can you guys see my thing? No, it's okay. You got it though. You got it at the end. It's a hard question. Okay, we're gonna talk about HL stuff now. Everything after this is HL. Although technically I feel like part of HL I already kind of discussed. I only need to really go into the initiation and what's it called? Post transcription modifications and stuff. Okay, so anyway, direction. So transcription moves in the 5' to 3' direction, where the RNA polymerase adds the 5' end of the free RNA nucleotides to the 3' end of the growing mRNA molecule. So it occurs from the 5' to 3' directions. Are you having issues hearing me? Are you good? Yes? No? Yeah? Good. We're good? Okay. Experiments and stuff will happen 4:30-ish. Well, my time. Sorry. In another hour and a half or so. Probably. If that makes sense. Someone asked. Okay, cool. Yeah, so transcription happens in the 5' to 3' direction, where it is essentially because the RNA polymerase will add 5' ends of the new RNA nucleotides to the 3' ends of the mRNA molecule. So, for example, the DNA strand is for the sense strand is from 5' to 3'. The DNA template strand will be 3' to 5', but the RNA will be created from 5' to 3'. Does that make sense? Yeah, 5' to 3' direction is transcription. Translation also happens in a 5' to 3' direction, where the ribosome will bind to the mRNA here and move in this direction, and it will translate each codon into an amino acid until it reaches the stop codon, where it happens from a 5' to 3' direction, and protein, the polypeptide, will be created here. So just know it's 5' to 3'. You don't need to know that it's why it is the way it is. It just is. I will be pinging you when we move to other topics as well. This is just because it's still the same topic. I will. Okay, cool. Transcription factors. So there are transcription factors in both prokaryotes and eukaryotes. So in prokaryotes, there's the promoters and the repressors. So the promoter is basically a section of DNA at the start of every gene, or right beside, adjacent to every gene, where transcription is initiated. So this is where the RNA polymerase will bind and start transcribing. Repressor proteins will bind to the promoter and prevent transcription. So that's basically what controls whether or not transcription happens, and therefore dictates whether or not that specific gene is expressed. Okay, let me just, it's from Sakura. Okay. And then in eukaryotes, there are similar transcription factors, but it's a little bit more complicated in eukaryotes. In eukaryotes, instead of directly, the RNA polymerase cannot directly attach to the promoter itself. There have to be these things called transcription factors, which are proteins that will bind to the promoter, which will then allow the RNA polymerase to bind to the promoter. The proteins have to bind first, and then the RNA polymerase will be able to bind to the promoter. Some transcription factors also have to be activated by the binding of a hormone or other chemical signals, and repressor proteins do still exist even in eukaryotes, where they will prevent transcription. So the diversity in the base sequences of the promoters and the sites to which transcription factors are bound is what results in a cell being able to switch on some genes and turning off some genes, because then basically the cells can decide that a repressor protein will bind to a specific let me explain this in this way. So in each cell, there are certain repressor proteins that will bind to very specific promoter base sequences, and that will be the case because then in that way it's controlled which genes are expressed and which genes are not expressed, because the repressor protein will only bind to very specific promoter groups, and those specific promoter groups are different in each for each gene, and the repressors that are present in each cell are different for each cell. And this is how specialization happens, where different types of cells will have different types of repressor proteins, and therefore bind to different bind to the promoters of different genes and prevent transcription of those specific genes. There we go. Okay, so for example, this is just from the thing again. So this is the template strand and this is the sense strand. The promoter is present upstream of the gene, since then the RNA polymerase can then move across the thing. And then the TATA box is basically thymine, adenine box. That is where the transcription factors will bind, and therefore allow the RNA polymerase then to bind to that area of the promoter and then begin transcription of the gene area. Yeah, for RNA transcript to happen, it has to happen. The start point for transcription to happen, the RNA polymerase has to join to the promoter, and for them to be joined to the promoter, there has to be certain transcription factors. There we go. Polyatyl and methyl cap is the next thing, which is why is this like this? Anyway, which is the post-transcriptional modifications. But first, we're going to talk about non-coding sequences. So each gene, basically in our DNA, there are thousands of sequences of bases that will code for proteins, and there are so many sequences that do not code for proteins. But some non-coding sequences will still have important functions. So the first is regulating gene expression. So some base sequences are sites where proteins can bind that either promote or repress the transcription of an adjacent gene. So again, like promoters and repressors, that's where they regulate gene expression. That is a non-coding sequence. And then there's introns. So in eukaryotes, there are introns that interrupt the coding sequence, which are removed from mRNA before it is translated. Introns have numerous functions associated with mRNA processing. Introns allow for smoother processing, essentially. And then there's telomeres. So telomeres are repetitive base sequences at the ends of each chromosome. The reason telomeres exist is because when replication happens, the end of the DNA molecule cannot be replicated. So then if there were no telomeres, that small section of the base sequence would be lost. The telomere will prevent the important part of the genes being lost and only remove the telomere areas, and that's just not transcribed, essentially. And then there's genes for tRNA and rRNA. So not mRNA. There is genes that code for tRNA molecules and rRNA molecules that are transcribed. So transfer RNA and ribosomal RNA that are both used for other purposes, not directly for coding for proteins, essentially. In terms of silencers, it's part of regulating gene expression. rRNA is ribosomal RNA. It's RNA that's used to synthesize ribosomes, I believe. Let me double check. Ribosomal RNA. Actually, I think we deal with ribosomal RNA elsewhere, no? I don't remember, but I think it's somewhere. Yeah, silencers were not mentioned directly in the textbook, but they're just part of regulating gene expression, as far as I know, for Wolfgang. Okay, so now we're going to talk about post-transcriptional modifications. There are two main ones that we need to know. Two main categories that we need to know. The first one is, I should start off with saying eukaryotic cells will modify mRNA after transcription, before the mRNA leaves the nucleus into the cytoplasm to be translated by the ribosome. These modifications will happen. There are two main types. One is the removal of nucleotides within the transcript, and one is the addition of nucleotides to the end of the transcript. So the first one is, as we said, adding. Okay, so we're going to talk about adding first. Adding extra nucleotides to give each end of the mRNA a special structure. So there's the 5' cap and the poly A tail. The the 5 prime cap, I was going to say 5 crimes. Okay, 5 prime cap is basically a modified nucleotide which is added to the 5 prime end of the RNA in order to it's basically, the structure is similar to ATP, where it has 3 phosphate groups and the base is guanine with an extra methyl group added. This stabilizes the 5 prime end essentially. And the poly A tail will be added to the 3 prime end, which is basically between anywhere from 100 to 200 adenine nucleotides, which are added to the 3 prime end of the RNA. And when translation happens, the translation will end right before the ribosome will reach the poly A tail. Because, as we know, translation happens from the 5 prime to the 3 prime end. The 5 prime cap will essentially stabilize these beginning, and the poly A tail will indicate where the ribosome is meant to stop, because obviously there's no it just doesn't translate past that. The stop codon will be right before the poly A tail. And the function of both of these is essentially to stop its digestion by nuclease enzymes, because when it goes from the nucleus to the cytoplasm, it has to go through the nucleoplasm, and it is a protein, so we don't want it to be digested. So that's why we have a 5 prime cap and a poly A tail. Okay, the next modification is introns and exons essentially. So like I said before, there are introns in mRNA sequences, in genes in general, and these introns will then be removed from the mRNA before it's translated, since we only want to have the important parts being translated into a protein sequence. You don't want the introns being translated because they don't supply any benefit really. So the introns are removed before it is translated, and the remaining parts are called exons, which are spliced together to form mature RNA. So the way it works is the DNA inside the nucleus will transcribe into a RNA transcript, which will then have a 5 prime cap and a poly A tail, and then the introns will be removed and digested, and the exons will then be linked together all together. And that's the only important part of the gene essentially, which will code for the proteins, and that will be called a mature mRNA. And then that mature mRNA will exit the nucleus, that's the nucleoplasm, via a nuclear pore, and the ribosomes in the cytoplasm will then translate the mRNA into proteins. So transcription happens in the nucleus, translation happens in the cytoplasm. So after transcription happens, the mRNA has to travel through the nucleus out into the cytoplasm for the ribosomes to do translation, and you don't want it to be digested by the nuclease enzymes in the nuclease enzymes in the nucleus. So that's why the 5 prime cap and the poly A tail exist, because it has to move from the nucleus to the cytoplasm. Anyway, does that make sense, guys? Translation occurs in the cytoplasm. Yes. Translation happens in the cytoplasm, transcription happens in the nucleus, because DNA is present in the nucleus. And ribosomes are in the cytoplasm. Okay, are we good with post-transcriptional modifications? Questions? No questions? Adding of nucleotide happen in pro and Eukaryotes. I think this is just eukaryotic specific. This is not prokaryotic specific. This is both eukaryotic, I believe. Because in the thing it just says eukaryotic cells modify mRNA. You don't need to know if prokaryotes modify. Which, I mean, they wouldn't, because prokaryotes have naked DNA, so there's no real modifications that are needed in that sense. For the first part, anyway. Okay. Questions? No questions. Oh, so what it could break down is the RNA. Yeah. Like, you don't want to lose any of the RNA sequencing. So you add a cap and a tail. It also stabilizes it in general, so yeah. I'm waiting. Wait. It's just, okay, so when I say stabilize, it's just the same thing. It stabilizes the ends where it protects them from digestion. It's the same thing. Like, it's just, if that doesn't happen, it's gonna get digested. I just say stabilize because that's the word they use in the textbooks, but it's basically just, like, protecting them. Okay. Are we good with post-transcriptional modifications? Because I know this was people have been asking for this one since I was doing DNA replication last week. Good? Yes? No? Maybe? Perchance? Perhaps? Okay, good. Cool. Now we're going to talk about initiation. Half of this I already discussed when I was talking about, what's it called? Polypeptide elongation. It's basically the same thing. You just have to know the fact that there's initiators. So an initiator tRNA with the anticodon AUG will bind to the small subunit of the ribosome. The initiator tRNA will carry the amino acid methionine. The reason the initiator tRNA is AUG is because the start codon on the mRNA is UAC, which is both the start codon and also the codon for methionine. So methionine is always the first amino acid in every protein polypeptide. The small subunit of the ribosome and initiator tRNA attach to the 5' terminal of mRNA and then move along the mRNA until they reach the start codon. The anticodon of the initiator tRNA and the start codon pair up by formation of hydrogen bonds. We know this already. The large subunit of ribosomes will bind to the small unit with the initiator RNA in the P site. So the initiator tRNA will be in the P site to begin with. The E site and the A site will be vacant. A tRNA with the anticodon complementary to the next codon will bind to the A site, a rival. The tRNA will carry the amino acid that corresponds, obviously, to the codon, and then a peptide bond is formed. I've explained all of this already, so I'm just going to really quickly go through it. This is translation. It's just, you just have to, the only part that's HL here is the initiator codon, but I've also explained that in the SL section for some reason. Yeah, so a peptide bond will be formed between the tRNAs in the P and A sites. The A site will then hold the dipeptide, and then the repeated cycle of events will begin here, where new amino acids are then added and polypeptide is elongated, and of course the ribosome will move along the mRNA, the mRNA, which will cause the tRNAs to change position. So the tRNAs will move from A to P to E, A to B to E, and so on and so forth. The P site will always have the main polypeptide and the E and A will usually be vacant as they're moving, and then when the new codon is exposed, the new tRNA will come into A, and then P, and then E, and I've already explained this, so I'm not going to do all of that again. But basically, for example, this is the small subunit, that's the initiator tRNA. They're going to scan along the mRNA until they reach the start codon. And then when they reach the start codon, the large subunit will come and dock with the small subunit, and this initiator tRNA will be attached to the P site. And then from the AUG start codon, you will keep going with the normal codons until you reach the any of the stop codons, and that's where translation will end. Yes, that is translation. Okay, so there are 3 binding sites in ribosomes, and all of them have different functions. And during elongation, every tRNA will briefly occupy each site. So there's the A site where the initial binding will happen. Hold on. Yeah, where the original binding will happen, where the tRNA will arrive carrying an amino acid. The P site is where the tRNA will move after it's carrying the growing polypeptide. The polypeptide will be transferred to the tRNA at the A site, and then that tRNA will then move back to the P site, and so on and so forth. And then E site is where the tRNA moves when it has no polypeptide when it's no longer holding the polypeptide, and then the tRNA will dissociate from the E site and return to the ribosome when an amino acid has been linked to it again. Therefore, the E site is known as the exit site. Okay, now we're going to talk about, before I talk about modification of polypeptides, there's also, you need to know about alternative splicing, although it's a really, really short thing that they've said about it. So basically, some genes have many exons, and different combinations of them can be spliced together to produce different proteins. This increases the total number of proteins an organism can produce from its genes. That's all it says on the thing. Okay, now we're going to talk about modification of polypeptides. Are we good so far? Are there any concerns? Because I know I rattled through that really quickly, but I just figured I'd get done with it as quick as possible. Because then we're going to finish this, and then we'll take a longer break before inheritance. Repeat which part? The alternative splicing? Okay. See, so it just says, some genes have many exons, and different combinations of the exons can be spliced together to produce different proteins. This increases the total number of proteins an organism can produce from its genes. Because when, for example, you're doing this, the exons can be spliced in different combinations, which will change the order of the codons, which will therefore change what proteins it codes for, therefore increase the number of total proteins. Is that in the guide? Yeah, I think so. Binding sites tend to not appear in the marking scheme. I mean, you need to know what they are anyway. You should. If you're HL, you should know what the binding sites are anyway. I can't confirm or deny whether or not they tend to appear in the marking scheme. I haven't properly looked at every single marking scheme. I'm not a teacher. I'm just here. But yeah. Okay. Parasite. Yes, I can. I mean, I'll be sending these slides anyway, but yeah. Are we good? This is what I'm going to talk about now. I haven't talked about this yet. Okay, so we're going to talk about modifications of polypeptides. There are 4 main types of modifications that you can do to a polypeptide. The first one is changes to the side chains. Second one is folding the polypeptide into a tertiary structure. Third one is excising part of the polypeptide. And the last one is combining 2 or more polypeptides or other components into a quaternary structure of a protein. So all of you should know, this is HL. All of this is HL. What was I going to say? You should all know the 4 structure, like the primary, secondary, tertiary, and quaternary structures of proteins. I'm not going to explain that. You should know that from proteins. But yeah, so this is an example given. It's insulin. So the insulin gene, when it's transcribed, is a 110 amino acid polypeptide. When it's transcribed and translated, it turns into a 110 amino acid polypeptide, which is called proinsulin, which will then be converted into proinsulin by a protease, which will remove 24 amino acids from one of the terminals. The remaining 86 is called proinsulin. That proinsulin will then be folded into a tertiary structure and remove 33 amino acids again to leave 2 chains of 21 and 32 amino acids. Those 21 and 32 amino acids will then then 2 amino acids will be removed from those 21 and 32 to result in a total of 51 amino acids, and that's insulin. So basically, all of that yap is to say, basically, this is a good example of the modifications that happen to these polypeptides, because in pre-proinsulin there are 110, right? And then that dark part gets removed, which is 24 amino acids, gets removed to form proinsulin. Proinsulin then folds into the tertiary structure. And then from proinsulin, that yellow part, which is basically 33 amino acids, which is removed, and then the thing that's left behind is technically the insulin after 2 more amino acids are removed to form a total of 51 amino acids. Let me double check with the guide whether or not you need to know the insulin step specifically, or if that's just an example, but I think you need to know it. Let me double check. Let's see, the new guide, first assessment 2025. What topic is this again? D point. I think let me double check. I don't know how much you technically need to know. I need to actually look at the guide and double check whether you need to know certain things. Okay, this is D1.2. Transcription, no, this is HL, yeah. So yeah, you need to know the 2-stage modification of pre-proinsulin, unfortunately. It says it on the thing. This is from the guide directly. So it says you should know that polypeptides are modified before they can function, and then yeah. So, okay. Pre-proinsulin has 110 amino acids. 24 of those amino acids are removed to form proinsulin. Proinsulin is formed when, after the 24 are removed, a tertiary structure is formed. After that tertiary structure is formed, 33 amino acids are removed, and then 2 more are removed to form insulin. I don't know if you need to know the locations. Oh, sorry. Okay, I will slow down. Apologies. There's 110 amino acids in pre-pro insulin. 24 amino acids are removed, and the rest of the 84 are folded 86 are folded into a tertiary structure, which is called proinsulin. From that tertiary structure, 33 more 33 amino acid sequence is broken again, and then there's two chains that are held together by disulfide bonds, out of which two amino acids are then removed to lead to a total of 51 amino acids at the end. Let me also just hold on. Give me a second, guys. Let me write this down. Okay, so pre-pro, oh my god, I can't spell. Pre-pro insulin has 110 amino acids. Okay? And then, oh, do not look at that. Hold on. None of this makes sense. Okay, yeah. Pre-pro insulin has 110 amino acids. Okay? And then we've got, sorry. And then we've got proinsulin, which is basically 24 sequence to leave 86 amino acid chain which is folded, and 3 disulfide bonds are made to stabilize the tertiary structure. And then insulin is when 33 amino acid sequence is removed. Leaving 2 chains of 21 and 32 amino acids held together by disulfide bonds, and then 2 amino acids are removed to yield mature insulin with a total of 51 amino acids. Okay. Kind of, if that makes sense. That's a lot of, yeah. But also, like, in the textbook thing, it also has where it's done and by what, but I don't know if you need to know that much. I can't lie, we didn't need to know this much. I don't know why they're making you do this. I feel bad for all of you right now. Like, why would they make you do this? Why would they make you do this? I don't know if you need to know all the numbers. In the guide, all it says is that you should know you should know the pre-pro insulin into insulin as an example. So if you don't know the exact numbers, I don't know if they would punish you for it, but you should know how it's converted, basically. Let me also in this text add, hold on. Okay, so the 24 amino acids are removed by, thing is, because it also gives you the terminals and everything, but they don't explain how that works. So I don't know what they expect from you. Like, this is insane. And the Golgi, this is actually like a little bit insane in my opinion, but again, I fear I don't make the syllabus for this subject. I would say general idea is you need to know how it goes from pre-proinsulin to proinsulin to insulin. Whether or not, like, you need to know the exact numbers, but you need to be able to appreciate that the way it works is from pre-proinsulin to proinsulin, there's removing part of the polypeptide, there's also folding of the polypeptide. From proinsulin to insulin, again, you're removing part of the polypeptide and you're changing the you're excising part of the polypeptide. So the point of the example is that there are modifications made to polypeptides, and this entire process essentially shows you the different types of modification. That's what you need to know. I will quickly go on to this, and then we will be done, and then we will take a long, nice 15-minute break before we go into inheritance. It goes from pre-pro-insulin to just insulin. It goes from pre-pro-insulin to pro-insulin to insulin. This thing. Pre-proinsulin to proinsulin to insulin. It's okay. Okay, so amino acid recycling. Now, this is fairly easy. There's this thing called proteasomes, which are organelles that digest very specific proteins. These proteins that need to be digested are tagged with markers called ubiquitin, whatever. You need to know the spelling, you don't need to know how to say it. Tagged proteins are recognized by regulatory subunits at either ends of a proteasome. And those regulatory subunits will recognize which proteins need to be broken down. A second subunit will then unfold the polypeptide and feed them into the central chamber, where the active sites of the proteases will digest them into short chains of amino acids. These short chains will then get passed out of the proteasome and get digested in the cytoplasm, which will yield in separated amino acids, which will then be reused for synthesis of new proteins and get attached to tRNAs. So the point of proteasomes is to digest the proteins that are damaged or have become functionless or are present in greater quantities than needed in order to create amino acids that are that can be reused. Does that make sense? That's everything for this part. I mean, everything for protein synthesis, I believe. Like, literally everything from start to finish. Questions? I will stop recording.